In this lesson, we're looking at sampling techniques. The first aim is explain why sampling techniques are useful, then describe different methods of collecting organisms, and finally explain how to use quadrats to estimate population size. A sampling technique is a way of recording and comparing data on organism distribution. Scientists are seen as people who go through painstaking lengths to record things in as much detail as possible, but where they can cut corners, they will. For example, if I wanted to determine the number of people in this picture, I could do it in two ways. One, I could actually count them individually, but that would take a very long time. Or two, I could take a few samples and work out an estimate of the population from those samples. I could also use sampling techniques to, let's say, work out or estimate the number of tuba players in this picture. You see, environmental conditions in habitats can vary, and this can have a direct effect on population size. Population is the number of one species living in a specific area. So, environmental conditions can vary in terms of temperature, in terms of light intensity, in terms of acidity of soil. All these things directly affect a population. To find out to what degree these factors affect a population size, I'd have to work out the population of, say, one species in two areas where one of these factors varies. For example, one field may get more light than another. How does that affect the population of sunflowers? To understand how these factors affect organism distribution, we must do two things. Firstly, measure the environmental factor we are investigating. So we could use a thermometer to measure temperature differences, or a light meter to measure differences in light intensity, or indicator solutions to work out the acidity or alkalinity of a soil sample. The second thing we must be able to do is determine the population size of the species we are investigating. This is where sampling techniques come into play. So if you understand that sampling techniques can be used to determine how a specific environmental factor affects a population size, then you understand aim 1. So that is why sampling techniques are useful. So firstly, we need to use methods to actually collect the organisms we want to count and compare. Let's first look at methods of collecting ground insects. One method is using pouters. Pouters are plastic containers that are transparent. They have a lid which seals the container and two rubber tubes that come out of the jar. One of those rubber tubes has a wire mesh underneath. This is the tube you will suck from. Another tube has no mesh, allowing insects to easily travel through the tube. So all you do is place your pooter over the ground insect and then you suck. You would then collect organisms in two different areas where the environmental factor varies, and you would count and compare the organisms. Another method for collecting ground insects is using pitfall traps. To make a pitfall trap, you dig a hole in the ground, you put a plastic container inside that hole, you put two pebbles around the hole and balance a lid on top to stop rain coming in. The gap is important so that insects can still crawl into this space. So you leave the container overnight, and what happens is insects go in and they slip into the container, but they can't get back out because it's slippery. And once again, you'd count and compare the organisms caught in two different areas. But not all organisms live on the ground. Some, for example, live in tall grasses. To collect these, we can use sweep nets. Sweep nets are basically nets which have lined with a cloth to stop things falling through. You would simply sweep up the organism you want to collect. You do this in two areas and compare what you find. Remember, each time, two different areas, count and compare. Some organisms live underwater. For these, you'd use a pond net. This time, your net is not lined with a cloth, so the water can get through. So you literally dip your net into the pond and scoop up the organisms. Once again, you do it in two different areas, count and compare. In exams, these usually appear as multiple choice questions. For example, and say, which one of these would you use to collect insects in long grasses? And you would say, sweep nets. Although I can imagine a 6-mark question where it asks you to describe different sampling techniques and you just list these different techniques and what they're used for. Remember how they're used, collect in two different areas, count and compare. And that's how we describe different methods of collecting organisms. Finally, we're going to look at how we can use quadrats to estimate population sizes in two areas. So firstly, a quadrat is a small metal square that we use to randomly place in an area and count the organisms we find inside. So let's say in this example that a nuclear power plant has recently opened and environmentalists are concerned that this might be impacting organisms nearby. Let's say in this example we're focusing on the daisy population in fields. So we would take a sample of daisies in one field near the power station and another field away from the power station. It's important that other environmental factors are similar, such as light intensity, soil pH, temperature. Otherwise, we won't know what's truly affecting the population size. Perhaps it's nuclear waste, but perhaps it's something else. The first thing you do is divide the field into a grid using a tape measure. Let's say this is a 10 by 10 meter field, so 100 meters square. So here I've divided my field up into 1 by 1 meter square, and there are 100 of them because it's 10 by 10 in each field. Next, I have to randomly pick coordinates to avoid bias. You see, I can't just fling my quadrat anywhere, because, for example, if I wanted to prove that nuclear waste is damaging the field, I might be drawn to looking at regions where there are fewer daisies, even though that's not really fair. I need to make sure that the quadrats are placed completely randomly. So I could pull numbers out of a hat, I could use a computer program to randomly generate numbers, and here you can see my randomly generated coordinates. So let's say out of my hat I picked a 3 first and randomly picked a 2 for the y-axis. So this is the x-axis, this is the y-axis. So what I do now is I place my quadrat at that coordinate. So it says 3, 2 over here. So I have to basically go 3 across and 2 up. It's actually the lower left corner of the quadrat that is used to pinpoint the coordinate. So then I'd count the number of daisies. So in my first quadrat I have 1. Now remember, the idea is I don't want to count every single square, just a sample. So I'm going to pick 10 random squares. So now we're moving it to 9, 1. So I go 9 across and I go 1 up. And again I've got 1 daisy. You'll notice that this daisy is kind of in between 2 squares. If the daisy is touching the rim of the quadrat, then you can count it. And I continue this process, so next will be 7, 5. So 7 across, 1, 2, 3, 4, 5, up. And now it's a bit trickier because you can see I've got 1 daisy there, but it is touching another one, so I could count that one too. So 2 daisies. So I record 2 there, and I do this until I finish the table. So what I've done so far is place the lower left corner of the quadrat at specific coordinates and counted the number of daisies in each quadrat. I've then repeated this 10 times. The number of times you repeat it really depends on the size of the field. If it's absolutely massive, you may want to take more samples. Now I need to calculate the average number of daisies per square. So firstly, I add up all these numbers here, and that will give me a figure of 20. So the total number of daisies I counted was 20. And then I divide it by the number of samples taken. So I took 10 samples. So divide that by 10, and that gives me an average of 2 daisies per square. So remember, total number of daisies counted divided by the number of samples, 10. And now I just multiply that answer by the area of the field. So I know that I've got 100 meter squares in this field, so I times 2 by 100. That gives me an estimate of 200 daisies in field A. I then repeat that with field B and compare the numbers. If field A had significantly lower numbers, but the environmental conditions were very similar, except for the fact that one's near a nuclear power plant, then I can assume that this power plant is having a negative impact on the daisy population. One other thing we can do with quadrats is use a belt transect. That's used to see how organism distribution changes across an area. A coastline is an excellent example of this. The first thing you do is mark a line across the area, so I've marked my line here. You would then place your quadrat at the start and count the number of different organisms. For example, 5 barnacles, 1 crab, etc. You then repeat this at regular intervals, every 2 meters along the line, for example. So I just move my quadrat along by 2 meters and count again, and so on and so on. Notice this time, because we're actually trying to see how organism distribution changes, we do not need to work out an average for a belt transect. We do want to actually see change. We're not trying to estimate a population. And that's how we explain how to use quadrats to estimate population size.